All right, welcome back. Hope everyone enjoyed that bit of a break. Now we'll move right along to our next case, an in vitro BE case study with Dr. Gajendiran Mahadevan, who's actually rejoining us from yesterday's workshop presentations as well. Just another reminder on Dr. Mahadevan's background. Dr. Mahadevan is a pharmacologist who joined the agency as an FDA Commissioner Fellow in the Center for Veterinary Medicine and then later moved to OSIS. He has been involved in the Bioresearch Monitoring Program for conducting inspections and alternative inspections as well. And so please join me in welcoming Dr. Mahadevan.
Hello everyone. Thank you for having me this afternoon. I am Gajendiran Mahadevan from the Office of Study, Integrity and Surveillance. I am very excited to share and discuss with you a couple of in vitro case studies this afternoon. On the first day of this workshop, you learned about an overview of in vitro BE studies presented by Dr. Monica Javinia and the inspectional approach for in vitro BE studies presented by Dr. Shripal Mada. The information you learned from these two presentations will assist you in solving the in vitro case studies I'm going to present to you today. This is an usual disclaimer. This presentation reflects my views and should not be construed to represent FDA's views or policies. Case study number 1. I'm going to present to you an in vitro drug release rate study conducted at a contract research organization and provide you some background information before asking a couple of questions for this case study. Let me go over the expectations for conducting a comparative in vitro drug release rate study. The study should be performed on at least 3 exhibit batches of the test and reference drug products to be tested, and testing 12 units of each of the test and reference drug products. An acceptable test method is developed followed by validation of the method before conducting the study. Comparative in vitro drug release rate study is a bioequivalence study and should meet regulatory requirements of 21 CFR Part 320. Reserve samples of investigational drug products used for the conducted in vitro B study should meet regulations of 21 CFR Part 323.8 and 21 CFR Part 326.3. And of course, the accountability of the investigational drug product used in the study should be maintained at the site. An in vitro drug release test was developed and validated using a USB apparatus in a contract research organization for an injectable drug product for comparative in vitro drug release rate to support a bioequivalence study. The drug product was an oil in water emulsion, and the surfactant was used to enhance drug release. The dissolution was validated with various percentages of surfactant to test the drug release from the investigational drug products. The site used 2.5% surfactant using the comparative drug release testing of 3 batches, each of test and reference drug products. Pre-study validation was not demonstrated at control conditions, and also the use of 2.5% surfactant was not part of the pre-study validation, nor was a justification provided. The following are the dissolution method conditions. USB apparatus type 2 was used for the study. The study was conducted at 37 degrees Celsius with 50 RPM. 1 gram of sample was used for the dissolution study. Phosphate buffer with pH 7.4 was used for this dissolution study. 500 ml of dissolution media used and 5 ml of sample was collected for sample analysis. Dissolution studies were performed with 12 units of each of the 3 batches of the test and reference drug products. Dissolution samples were collected at 5-minute time intervals for 6 time points: 5, 10, 15, 20, 25, and 30 minutes. The collected study samples were analyzed using validated HPLC method with a UV detector. The percentage of drug release for each sample was calculated. Question number 1. Based on the provided information in slides 5 through 7 for the in vitro comparative drug release study, if you were assigned to conduct an inspection of the study at the site, what are the elements you find deficient and why?
And bear with us momentarily. We're checking on some technical issues we're having with Gaja connecting. Gaja, we see that you're unmuted in the Adobe Connect room. Now you're back on mute. Is it possible that your microphone is double muted, perhaps? Go ahead and give it another try there. And since we're still not getting anything, Gaja, if you can go ahead and check the microphone settings, the little white arrow next to your microphone, to check to select that you have the microphone, has the blue check mark next to it you want to use. Go ahead and try speaking again. All right, we're still not hearing anything from you, even though it's showing your mic as disconnected. Can you possibly try choosing a different microphone on your PC? When you click on that settings arrow to select your microphone, perhaps there's another microphone on your computer you could choose. If you're using the microphone, the only one that's on your PC, you may try using Alt F4 on your keyboard to get the local mute off. We do see that you're unmuted in Adobe Connect, but most likely you're still muted locally. Either your built-in microphone, and again, if you can check to see if the microphone settings, so up there next to your green microphone, the little white arrow, if you click on that and select your microphone, check to make sure that the microphone next to the microphone, or the microphone you want to use, has a blue check mark next to it. Again, and I see you're back off mute again. Go ahead and say something. We're still not hearing anything. All right, and I apologize to the audience. Please bear with us. We're going to get these issues ironed out here quickly. Gaja, let's try something crazy. I want you to go out and actually exit and come back into Adobe Connect entirely. I'm
getting a little echo now. Maybe one of the things that we could have a little conversation about is the discussion about guidances, right? So that is, I saw that a little earlier on, what guidance should be being followed for the method validation for the in vitro studies. And there is the suggestion of the ICHQ2R1, and as mentioned, it's not a bioanalytical method guidance, but it is a chemical method. This is the reality with the in vitro studies, that the crossover with the guidances between for bioanalytical analysis versus for just strict analytical analysis gets a little blurry. But for the in vitro BE studies, we do often just want to make sure that the guidance used is as fit for the analysis as possible. And so it's best to just ensure that the analysis you're going to be doing is validated appropriately. I think that Shri Paul, Dr. Mata yesterday was talking a little bit about this, and there's a little bit of the need to sort of tailor your approach as necessary, given the additional weight that an in vitro BE study might be used in terms of a drug approval. That it's important that the validation is sufficient to be clearly reliant on the data generated. Gajo, is that you?
Yeah, do you hear me, Sean?
Excellent. Now we can hear you. Yes, sir.
Oh, okay. Thank you, guys. I really appreciate it. I don't know, something, technology always gives trouble when you need it. Thank you so much, guys. I believe you got sufficient time to go over the question number 1. So I'm going to read out to you all. Question number 1, based on the provided information in the slides 5 through 7 for the in vitro comparative drug release study, if you were assigned to conduct an inspection of the study at the site, what are the elements you find deficient and why? Let's keep chatting in the chat box. Your active participation is really important to get a maximum out of this case presentation. Please go ahead and type. Once again, I apologize for the technical snag. Let's keep going. Thank you very much. Answer to question number 1. The release profile of drug from the drug product or control condition was not demonstrated during this pre-study validation, and the justification for the use of surfactant to achieve sync conditions for the drug was not provided. The site did not demonstrate and validate sync condition at 2.5% surfactant. However, the comparative drug release rate study using the investigational drug product was conducted at 2.5% surfactant. For the next question, I'm going to provide you some background information followed by the question. So this slide provides the investigational drug products received at the site for both test and reference product. The test product used in the study were shipped to the CRO from sponsor in 3 shipments on March 10th, March 17th, and March 24th, 2021. The reference products used in the study were shipped to the CRO from sponsor in 1 shipment on March 31st, 2021. This slide shows you the accountability information for the investigational drug product used in the study. Please pay attention to column number 2, shipment dates, and column number 6, number of units remaining at the site. Before answering the question number 2. Question number 2. An FDA investigator observed objectionable condition during the inspection and issued a Form FDA 483 to the management at the inspection closeout. Based on the information provided in slides 11 through 12, what are the likely findings for the observation? Hello, everyone. Let's think about the question number 2. Let's start chatting in the chat box. So there are a lot of responses coming up, and please don't worry. We have plenty of time, so I will cover those comments and questions at the end. There are a number of comments on retention of samples were not maintained at the site for each shipment. Yeah, that's absolutely correct. Retention samples are not as per the FDA requirement. Shipment information is not adequate. Exactly, all quantity was consumed and no reserve samples. Yeah, person receiving the samples were not documented. Receipt and storage conditions were not provided. Yeah, there was a question about if the batch number is the same, should we retain each shipment? Yeah, that's sometimes very important because someone pointed out in the chat box that the storage condition when it was arrived. So it's always the shipment, though it's a different batch number, if it's arrived in different shipment, it's better to keep a reserve sample. Yeah, there is a question on three test product shipments were within short duration. But if it's an independent shipment, it's better to keep a reserve sample for each shipment. If it's all together, that's a different story. But if it's an independent shipment, so the site have to have a reserve sample for each shipment. Can we go back to the document? I don't know whether Jeff can show us the last slide with the different columns. Yeah, so it is displayed. Please have a look at it. So Delores Perry, please, the slide is displayed. So if you have any comments or questions, please go ahead and put it in the chat box, please. Thanks. Yeah, there is a question on guidelines to be followed for qualitative percentage decrease of pepsin activity in sucralfate suspension. I think it's covered. I believe yesterday I did participate in the panel discussion about the sucralfate product specific guidance. So if time permits, I will talk about that later end of this presentation. Okay, let's continue the presentation, and we will go over the questions and comments later. An FDA investigator observed objectionable condition during the inspection and issued a Form FDA 483 to the management at the inspection closeout. Based on the information provided in slides 11 through 12, what are the likely findings for the observation? Let me go over the answer to question number 2. Since it was a pivotal comparative in vitro drug release rate study using both test and reference drug products, reserve samples for the conducted study was required by applicable bioequivalence regulations 21 CFR Part 320, 38 and Part 320.6.3. Reserve samples are required for each of the shipments of the investigational drug product received at the site and that were used for the conducted study. Because there was no reserve samples, we were not able to verify the authenticity of the test and reference drug product used in the bioequivalence study. Okay, let us switch gear to the second case study on in vitro permeability testing. So the schematic diagram shows a gecko-2 cell monolayer with the apical and basolateral sides, and the keys shows the low permeability and high permeability markers along with the efflux marker and the study drug. What are the expectations for the in vitro permeability assay? The integrity of gecko-2 monolayer should be assessed before and after an experiment. Controls such as low and high permeability markers should be used for the in vitro permeability testing. For bidirectional gecko-2 assay, efflux markers should be used to demonstrate asymmetric permeability. By conducting the permeability assay, efflux ratio and apparent permeability are calculated. So this slide shows the information about the drug and the study. The drug used in the study was a PCS class 1 drug. Bidirectional gecko-2 assay was performed. PCS-based biowaiver was submitted for an abbreviated new drug application. No clinical bioequivalence studies were conducted for this. This was not a bioequivalence study. In vitro permeability studies were conducted using gecko-2 cell monolayers for drug Y to support biowaiver for a drug product. The bidirectional apical to basolateral and basolateral to apical permeability of drug Y was determined. Gecko-2 cell monolayer integrity was assessed by transepithelial electrical resistance. The permeability of low and high permeability markers and efflux marker were determined in the study. The site conducted analytical method validation for drug Y and not for the controls used in the study. Study samples were collected from the donor side for 2 time points, 60 and 120 minutes. Study samples from receiver side were collected for 4 time points, 30, 60, and 90 and 120 minutes. The collected study samples were analyzed by HPLC MSMS. The concentration of markers and the study drug were quantified for subsequent calculation of the apparent permeability coefficient. Question number 3. Based on the provided information in slides 20 through 21 for the in vitro permeability study, if you were assigned to conduct an inspection of the study at the site, what are the elements you find deficient and why?
Thank you, guys. You saw that question, and let's think about the answer for question number 3. Question number 3, please. The question number 3 is for the in vitro permeability testing. I did provide a background information for the question. So based on that background information, please please go over the question and let us chat in the chat box, and then we can discuss it. Exactly. The method validation did not include the controls. That's correct. The analytical method validation was not conducted for the controls. Though for the method, they used the controls. However, for analytical method validation, the controls were not validated. So the question is, what are the elements you find deficient and why? So let's find out what is missing there. So someone pointed out that the analytical method validation was not conducted for the control. That's exactly correct. The permeability markers and efflux markers were quantified in the study samples, but the analytical method was not validated during pre-study method validation. That's correct. Yeah, someone saying that having different sample time point at the donor and receiver site. Yeah, that was collected for different purpose. But the question here is, what is the major deficiency here that really throw the train out of the rail? So let's think about it. Okay, let's go over the answer. So then we can discuss, so you may get some idea.
If you were assigned to conduct an inspection of the study at the site, what are the elements you find deficient and why? Let me go over the answer to question number 3. The site validated the analytical method for quantitation of drug Y. However, they did not validate the analytical methods used for the quantitation of the permeability and the efflux marker from the study sample. Question number 4. Do you think not conducting pre-study validation for the permeability and the efflux markers in slide 20 has an impact on accuracy of the analytical results, and why?
Yeah, thank you so much, guys. Yeah, I think I started seeing the information, the comments on the chat box. Yeah, someone was saying that without validating the control, the data is unusable. Exactly. So the method was not validated for the 3 analytes, right? So the low and permeability markers as well as the efflux marker. The method only validated for the drug Y. So it's a yeah, last yesterday, there were, the number of presentations and methods should be validated for all the analytes. So it is very important. Yeah, the compromise, the accuracy of the result. Exactly. Yeah. Yeah, there was a comment on conducting concurrently the analyte. That's something, you have to consider. I remember I displayed in the one of my slides that, when you select, so your drug is for sure you are going to test. So make sure that whatever the low and high permeability marker you are selecting along with the efflux marker, please make sure that the compatibility, because there are number of low and high permeability markers are available in the BCS classification guidance. So please make sure do diligent background work on the compatibility. That is very important. So that will get rid of the compatibility issues when you analyze. Thank you. Yes, of course, you have the data for all 4 analytes because that information the site was generated from the study sample. However, the method was not validated for 3 analytes out of 4. So yeah, the data is not usable. Yeah, someone mentioned about linearity. Yes, that's also very important because, so during method evaluation, so those information have to be validated for the other 3 analytes, the low and high permeability markers as well as the efflux marker. Yeah, someone mentioned about the pre-study. I think I mentioned that, so whatever the method you develop, that has to be validated, because that is very important step. Though someone asked about whether the guidance is available for this particular drug, I don't want to go into detail, but as I mentioned yesterday during my panel discussion, so when you go for a bioequivalence study or pivotal study for in vitro study, so please go over the product specific guidance and also pay attention to the product you are choosing. That's very important because product specific guidance is available for different products. So you have to make sure that what product you are going to use it. So that's very important. Yeah, someone mentioned that the pre-study validation is not conducted for 3 analytes, so the data become, not valid. Exactly. Okay, let's go over the video, please.
Answer to question number 4. This in vitro permeability study was a pivotal study for the biowaiver application. Accurate quantitation of the controls, that is permeability and efflux markers used in the study from study samples using validated analytical methods was critical because the results of these controls were used to determine the acceptance of the permeability experiment and for establishing the permeability classification of drug Y. Therefore, this objectionable finding affected the acceptance of study data. This is my final slide showing the resources available for you to conduct in vitro studies. The first bullet is showing the website where you can find the dissolution methods. The second one is guidance for drug. The third one is the product-specific guidance for generic drug development. And then final one is the guidance for biopharmaceuticals classification system. So this is the new guidance published in 2021. Please have a look at it. Thank you, everyone. Have a nice afternoon.
Okay, thank you, guys. So let me go over the some remaining questions, so I can go over. Okay, there is a question on a CRO conducted a bioavailability bioequivalent study sample analysis. Can do the IVPT sample analysis is GLP approval is required. So for conducting the BE studies, GLP regulation is not required. However, the quality management system should be followed for these studies. Yeah, there was a comment on, so there was robustness of the methodology was not established for the 3 analytes. Yeah, that's correct. The reproducibility was not available for the other 3 analytes. That's correct. Okay, so there is a question on what is the rational use of surfactant. So as I mentioned in my background information that this is oil in water uh, drug products. So the site uses surfactant to enhance the release. So they did use different methods to try to conduct this dissolution study. So they were not successful, so they tried to use surfactant for conducting this study. The next question is, what is the guidelines to be followed for qualitative percentage decrease of pepsin activity for sucral suspension? Yeah, that is something I mentioned yesterday during the panel discussion. There is a product-specific guidance available for sucralfate suspension. There are 4 different methods needs to be conducted. So yeah, please follow that 2017 product-specific guidance for sucralfate. There is pepsin. Another one is that the human serum albumin or bovine serum albumin study assay, and the other 2 are the binding studies. That is a equilibrium binding and um, equilibrium binding study. Yeah, there is a question on what reference standard was used when the test product was run. Yeah, so as I mentioned in the beginning of my presentation, that the permeability study was not a bioequivalence study. So here they used only the test product. So there is no reference product. That's the reason I also mentioned that this is not a bioequivalence study. Regarding the reserve sample, reserve sample needs to be maintained for each lot. I mentioned about shipments, not lot. Though for convenience, there are three lots for each of the investigational drug product, but they send it by different shipments. So that was the key here. So if 2 lots sent by one shipment, if one reserve sample is available for 1 lot for that particular shipment, that's fine. But make sure that each shipment, the reserve samples are maintained at the site. That's very important. Okay, the next question is, if the samples from the first shipment is adequate for the experiment, is it acceptable that subsequent shipment is not open? Yeah, that's perfectly fine, because that's also I mentioned in my answer key that yeah, the investigational drug product received and used in the study, that's the key. So if the drug product is not used, one of the shipment is not used, that's fine. So as long as you have reserve sample for the shipment you used for conducting the study, you need to have the reserve sample. Okay, there is a question on where to communicate for the product specific guidance if any specific product specific guidance is not available. Yeah, I think this question also I addressed yesterday during the panel discussion. The product specific guidance is published by Office of Generic Drug in the CDER, in the Center for Drug Evaluation Research. There is a division in the Office of Generic Drug which takes care of the product specific guidance publication. Please reach out to OGD, that is Office of Generic Drug. So if you need any specific information for conducting a bioequivalence study for the drug product, the specific product specific guidance is not available for that particular drug product. Okay, there is a question on, in case where a client, a company may use an unvalidated method and the method used was within the validated parameters of the method. When it was validated, the data can be used to support bioequivalence? Yeah, for bioequivalence study, it's very important, again and again, this is something, we emphasize that the method has to be validated before the study sample analysis. That's very important. Even if Dr. Gopapis was mentioned that if some of the components maybe not conducted during method evaluation, for example, long-term stability study or freezer stability study, those stability studies where you conducted for only 3 freezer stability study, but during sample analysis you went beyond that validated number of freezer cycle. You can go ahead and conduct the freezer stability number of record freezer stability study, but the method has to be validated before conducting the sample analysis.
Okay, it looks like unfortunately that's all we might have time for. Thank you again, Dr. Mahadevan, for the great presentations. And despite just a few technical challenges, we had some wonderful discussion there.